Good day, everyone. So we're starting with the course enzymology. I'll be going through the definition of enzymology, classification of enzymes, the properties of enzymes, and terminologies we should get familiar with in enzymology. So yes, let's get into it. So, enzymology is the branch of biochemistry that studies enzymes and how they carry out their effect in relation to their structure and functions. Now, enzymes are a protein substance that helps catalyze reactions in a biological system. Now, enzymes and the chemical catalysts are similar to an extent, but now these chemical catalysts catalyze chemical reactions outside the biological system, while enzymes catalyze reactions within the biological system. But aside this, they share almost all the same properties of not being used up after the reaction, being able to catalyze another reaction after the first one it has catalyzed. Now, going to properties of enzymes, they have high molecular weight, they carry out their effects in your aqueous solution, and they have higher catalytical ability than just normal chemical catalysts. Now, they are also seen to reduce the activation energy required for a reaction to move forward. They speed up the rate of reactions. Now, these enzymes are also proteins, and they remain unchanged after a reaction and are able to catalyze another reaction after that, the same reaction after that. And they are proteins, so they have a specific temperature and pH they work under. Above this temperature they become denatured and not able to carry out their function. Now, these enzymes, they can also carry out reaction help in catalyzing reaction that either performs synthesis of a compound or lysis of a compound, in the sense that they can synthesize a compound from two compounds or more, and they can break down a compound into more than one component. So yeah. And lastly, enzymes are specific, having a specific substrate they carry out their catalysis upon. Now, under the properties of enzymes, I talked about them being able to perform lysis and synthesis. Now, looking at the two images we have, or the two figures we have here. Now, the first one shows a catalyzing reaction, while the second one shows a lysis reaction, breaking the bond between those two molecules. Now, if you also notice something, as I said before, they have specificity for their substrate. Now, the first one has a shape that can only fit in for the particular substrate. Looking at the second one, the shape of the active site is different from figure 1, so that is another difference and that thing that makes enzyme specific. Now, the mechanism of action of how enzymes do work. Now, they work by lowering the activation energy of a reaction. Now, looking at the first figure we have here, which is figure 3, we see that the first graph and then the second one. Now, the first one is the original reaction without it being catalyzed, without an enzyme catalyzing the reaction, while the second one down is the enzyme. When there's enzyme catalyzing the reaction, you see how the activation energy has been quite lowered, and now the free energy is the amount of energy that is being released. And then from that, you can see that this reaction is hexagonic because it's releasing energy into the surroundings, or is releasing energy. Now, the mechanism, it lowers the activation energy, provides an alternative pathway, that's one day the enzyme catalyzed one, and then reduces the reaction entropy. The reaction entropy change by bringing substrates together in the correct orientation to react. Now, these substrates, they could be freely moving within the solution, or within the environment, or within the biological system. But now these substrates brings them together and helps the reaction to take place quickly, and the entropy, that is the rate of disorderliness of the system, is actually lowered so that the reaction can get to completion quickly. Now we'll go to the classification of enzymes. There are 7 classifications of enzyme presently, but in the past there were just 6. But recently in 2018, the 7th one, translocase, was discovered. So now, a way to quickly understand or quickly remember this classification of enzymes is over the hill. Now, O there stands for oxidoreductase, then T stands for transferase, then H stands for hydrolase, and then now for the hill, for the ILL, do we replace the first I with the L, so it becomes LIL. So the first L is lyase, the I is isomerase, and the L is ligase, and then we have translocase. So now let's go into their definition and examples. Now, oxidoreductase catalyzes redox reactions, which are oxidation or reduction reactions, and their common name are either dehydrogenase or oxidase. Now, transferase catalyze group transfer reactions, such as the transfer of a glycosyl or methyl group, and they are mostly their common name or their is transaminase or kinase. Now, the hydrolase, they catalyze hydrolysis reaction, and then these are reactions that use water to break down. So an example is lipase and amylase. Then lyase catalyzes the formation of double bonds by the removal of groups, and then the common name under lyase is decarboxylase. Now, isomerase catalyzes the transfer of groups within molecules to yield isomeric forms, and their common name we see them with is isomerase or mutase. Now, ligase is the formation of carbon to carbon bond, carbon to sulfur bond, carbon to oxygen bond, and carbon to nitrogen bond, and these reactions involve or use ATP, and then their common name is synthetase and carboxylase. Now, translocase catalyzes the movement of ions or molecules across the membrane. Now, this catalyzes reactions where molecules from one part of the membrane need to be taken into the other part of the membrane. So an example of transferase is glucokinase that catalyzes the first reaction in the glycolysis pathway. Now we have for hydrolase, we have the lipase that catalyzes the cleavage of a fatty acid from the trialglycerol molecule to yield one fatty acid and one dialglycerol molecule. An example of ligase is pyruvate carboxylase that catalyzes the conversion of pyruvate to oxaloacetate, which is converted to malate and taken out of the mitochondria and converted back to oxaloacetate and then enters the gluconeogenesis pathway. And now an example of oxidoreductase is alcohol dehydrogenase, and then alcohol is being formed from pyruvate, which is the end product of the glycolysis pathway, and then this happens when under anaerobic conditions. Now, an example of a lyase is aldolase, and it catalyzes a reaction in the glycolysis pathway, as seen there. It catalyzes phosphate 1,6 biphosphate to dihydroxyacetone and phosphate and glyceraldehyde triphosphate. So here it breaks or it cleaves a bond and a double bond is being formed. An example of isomerase is phosphoglycerate mutase that catalyzes reaction that changes 3-phosphoglycerate to 2-phosphoglycerate. Figure 4 there just shows the glycolysis pathway and different enzymes that catalyzes reactions there. And then an example of a translocase is the ATP ADP translocase. Sorry for the typographic error. So it catalyzes the reaction and helps in transfer of ADP into the cell, into the ADP into the membrane, across the membrane, and then the ATP synthase converts that ADP to ATP, and then the ATP now is carried across the membrane, and this happens in the mitochondria. Now we're coming to the part that I term the terminologies in or common terminologies in enzymology. Now the first one is simple enzymes. These are enzymes that do not require any metal, ion, or any organic group to help in the catalyzing of a reaction. Now the first one here is the coenzyme. These are organic molecules that are bound to the active site of an enzyme and helps and aids the enzyme in catalyzing its reaction. Now catalyzing a reaction. Cofactors are metal ions that are bound to the enzyme and help the enzyme in the catalyzing of reactions. Now prosthetic groups are metal ions or organic groups that are tightly bound to the enzymes and help also in catalyzing reactions. And examples, we have the FAD, we have the biotin, we have the zinc ion, magnesium ion, and copper ion, and some others. Haloenzyme is a complete catalytically active enzyme that is bound to its cofactors and its coenzymes. Now, apoproteins is the functional or is the protein part of an enzyme without its characteristic prosthetic group, which means these also apoenzymes are just proteins. It's just the enzyme without cofactors and coenzymes, but they are specifically and more preferably called apoenzymes because there is apoproteins in high density lipoproteins and low density lipoproteins. So haloenzyme is simply the combination of apoproteins and the respective cofactor or coenzymes. Now this is the structure of a holoenzymes, showing the cofactor, the catalytic site, which is also the can also be called the active site, and then we have to show the coenzymes, and then the apoenzyme, indicating that the cofactor and the coenzyme is different from the apoenzyme. Now, the differences between coenzymes and cofactors. The major difference to note is that coenzymes are organic molecules, while cofactors are metallic ions. And coenzymes bind loosely to the enzymes, bind loosely with the enzymes, while cofactors bind either loosely or tightly to the enzymes. Now, examples of coenzymes are vitamins, while examples of cofactors are minerals. Biotin is a vitamin that helps in the transfer of CO2. It is seen as a coenzyme. It's seen as a coenzyme in the first step of fatty acid synthesis, which is the formation of malonyl CoA, which is the rate limiting step. So that is the end for today's video, and thank you for watching. Look forward to the next one. Yeah, that is it for today, and don't forget to like and subscribe. Thank you.